So good morning, I'm Brian Wandel. I'm also a Deputy Director of the Institute, and thank you for joining our annual symposium. It's my pleasure and honor today to introduce our next speaker, Professor Michael Kahana, who's the Edmund and Louise Kahn Professor of Psychology at the University of Pennsylvania. Professor Kahana will be speaking about really important and fascinating work that explores the principles of human memory function. And those mechanisms will clearly be related in the long run to the kinds of phenomena that Michelle was just talking about in terms of both neural and glial systems. And as you'll hear, Professor Kahana takes a really broad approach to the study of memory, including as the title of his talk is today, the measurements of electrical activity in the human brain. And he even seeks ways to intervene in the human brain to enhance memory. He also makes behavioral measurements, and he builds rigorous system models, meaning computational models, that aim to capture the principles of how memories are stored and retrieved. He's written an important influential book, The Foundations of Memory, and he's been recognized for his contributions by many awards, including the Trollin Prize from the National Academy of Sciences. Professor Kahana received his PhD from the University of Toronto in 1993, and then he went on to Harvard as a postdoc with a very distinguished memory researcher, Bill Estes. But in speaking with Mike to prepare for this introduction, and something that I think younger scientists all need to remember, Mike really spoke in very warm and personal terms about the next 10 years of his life, rising as through the academic ranks, at how important it was when he was at Brandeis for 10 years, working with and learning from Eve Marder, Larry Abbott, and John Lissman. And Brandeis has been a really remarkable source of new ideas and talent that integrate computational work with neuroscience. And Professor Kahana's work reflects that intellectual environment established by Eve and Larry and Lissman. And it's really something that I know we all on the executive committee seek to promulgate and establish and nurture here at Stanford also. About 15 years ago, Professor Kahana moved to the University of Pennsylvania, which, like Stanford, has a medical school, school of engineering, and basic science departments in close proximity. And with his technical breadth, his enthusiasm, his just bubbly nature, Professor Kahana has been able to take advantage of that environment. Also, has gone beyond to build an international network of collaborators, including some here, Anthony Wagner in psychology, Joseph Parvizi at Stanford, and just down the road, he works with Eddie Chang at UCSF, another good friend of our university here. Professor Kahana's work includes measuring and analyzing electrical signals. And I believe we'll hear something about some of the more prominent work that he has been doing, supported in part by Darpa and other grants, with the aspiration to develop a device that can both monitor and modulate different brain regions involved in the memory storage. So thank you for joining our symposium and for sharing your ideas. And I'm very much looking forward to hearing your talk. So thanks, Mike.
Thank you, Brian, for the very kind introduction. I hope everyone can hear me. It is an honor for me to address this esteemed group, and I look forward to a time, hopefully in the very near future, when we can all gather again in person. I want to pause for a moment before my presentation to acknowledge a giant in my field, in our field, someone whose work inspired me through graduate school and led to the development of my major theoretical work on human memory, the temporal context model. Although I never studied under Gordon Bauer, who, as you all know, passed away quite recently, I had many wonderful interactions with him early in my career that I'll forever cherish. And I want to dedicate this talk in his memory. May his spirit of kindness to others serve as a model for all of us. Maybe it would be helpful especially for some of the folks in the audience who are not working in areas that close to my field, to begin by sharing a little history of my own journey. I started training in the area of mathematical psychology, and I had really very little awareness of the human brain in my early stages of my career. In the area of human memory, mathematical psychologists didn't always think that the brain had any relevance to memory, and maybe some physical substrate for memory, but not clear that it really mattered. But our older siblings in the perceptual sciences had long recognized the importance of linking physiology, physics even, for example, of light and physiology to behavior. And so that research was always part of my training. I think it was, for the training of many of us, played a critical role in our intellectual development. So in the back of my mind, even before I ever investigated the brain, I always imagined some point where neuroscience would join up with cognitive theory to help us understand the mysteries of human memory. Today, I'm going to tell you about the electrophysiology of human memory. I'm not going to tell you about mathematical modeling of memory or cognitive theories that I've worked on. And every journey must have a beginning. So how do we define the beginning of the journey? It's not always easy to identify the first germ of an idea or of an impulse that led you down a road. But in 1997, while I was a junior faculty member at Brandeis University, as Brian mentioned, I ran into a neurosurgeon who invited me to speak to his colleagues about memory. And I share this because of the interdisciplinary nature of the Wu Tsai Institute at Stanford. Joe Madsen had studied engineering in college and became a neurosurgeon. And when he heard that mathematics was relevant to the study of human memory, and he treated many patients with memory problems, he invited me to speak to neurosurgery grand rounds at Harvard Medical School. And I won't bore you with the details of this long and maybe it's a little interesting story, maybe over dinner when I come out to visit you in person. But at that moment, I decided to venture into neuroscience without ever having taken a course in biology. And I had the great fortune of having senior colleagues who didn't mind teaching me basic stuff. And Eve Marder, Larry Abbott, and John Lissman were my early teachers in neuroscience. I want to mention one other thing that by way of introduction. Electrophysiology of human memory is an enormous space. There's no way I could possibly even attempt to cover the outlines in a brief presentation. When I was in college, I read a remarkable pair of books, not the whole pair of books, but many chapters. The PDP books that Jay McClelland and Dave Rummelhart and their team developed. And I think it's important for us to not only write journal articles, but also to try to put together knowledge in a way that students can grasp it and learn it. And so a lot of the broader scope of the electrophysiology of human memory will be addressed in a book that I'm putting together with several of my colleagues, Yusuf Eziat, Tung Fan, Christoph Weidemann, and Josh Jacobs. And I just put up an outline of the chapters that we've already drafted, but hopefully in a year from now, you'll be able to share that with your students and trainees. Well, the real story of how my research in this area kind of took off was by making a mistake. In the 1990s, the standard method for studying neuroscience in memory was to look at evoked potentials, changes in physiology that consistently happen after the appearance of a stimulus on the screen or an auditory stimulus that a person might hear. And when I first made the foray at Children's Hospital into doing human recordings in neurosurgical patients who had implanted electrodes, we didn't know how to synchronize the computer with the task. So our synchronization was flawed and there was jitter of a few hundred milliseconds. And that mistake led me to realize that brain oscillations are evident in these neural recordings, right? If you had averaged across trials, you might not have seen those brain oscillations. And I remember running with a printout. We used to carry around printouts into John Listman's office because he and Larry had been talking about theta oscillations, these 4 to 6, 4 to 8 hertz oscillations that in the rodent hippocampus appear prominently during navigation and orienting behaviors and learning and correlate with long-term plasticity and synaptic modification. And I had a theta oscillation, and I ran into his office and I showed it to him. And we wrote a number of papers. I wrote some with John, some with others. I did some work on perception and neural activity with Bob Sekuler. And that work appeared in the period of 1999 to 2003 and is summarized in one article I wrote called Theta Returns that summarized the evidence from human neurosurgical patients for a functional role for theta, that theta is not just a signal of some disease state of the brain. We then created a set of experiments called Yellow Cab. There were a whole variety of these video game-like experiments. Why did we create them? Well, Joe Madsen told me that the kids at Children's Hospital didn't want to memorize lists of common nouns. Okay, fine, I understand that. So we would have them play video games. And those video games, which I conducted a variety of with students and colleagues, including my colleague Yitzhak Fried at UCLA and others. I think Professor Parvizi was a trainee when we did those first studies in UCLA. We uncovered the role of play cells in the human hippocampus, Ekstrom 2003, goal cells, grid cells in 2013, and a variety of findings that basically simply showed this. They showed that the rodent model of neurophysiology as it relates to behavior and memory and learning in spatial environments translates to the human brain. There wasn't a lot of creativity, truly. It was just following the rodent experiments and finding the ways in which the human neural data were similar and different. I like data. I like collecting lots of data and analyzing it in different ways. And one of the things that we looked at quite intensively in the period of 2006 to 2009 was how narrowband oscillations and broadband spectral modulations together relate to and separately relate to neural activity of individual neurons. Just in a quick survey of the path that I took through this work before I get to some specific results that I'll tell you about in my more recent work. We then spent a number of years trying to understand these broadband phenomena. So we uncovered broadband phenomena. It turns out that high frequency broadband activity strongly correlates with the firing of individual neurons. That was finding made in rodents and mice, rats, and subsequently we uncovered the same relationship, a very strong relationship to the human brain. It has also been documented in monkeys. So we then thought, well, high frequency activity is great because you can use it to identify the spatiotemporal pattern of neural activity. Kind of instead of thinking about what's happening overall, you can think of how it unfolds over time during a task. Now, of course, in the background, there was always the big question, which is how do I make my mentors, both my direct mentors and my indirect mentors, people like Bill Estes and Gordon Bauer and Ben Murdoch, appreciate this neuroscience work. I was still going to the mathematical psychology meetings and talking about math models. And of course, I cared about that. So we began studying neural patterns and how they relate to cognitive theories. And there's a wonderful annual review paper by your own Anthony Wagner, good friend and colleague, that I always distribute in my graduate courses about how to study pattern representations in the brain and relate them to cognitive theories. And we did that with intracranial recordings, much like Anthony and others have done with fMRI recordings. Now, on again and off again, we wondered about modulating the brain with stimulation. Clinically, neurologists stimulate the brain to try to identify functional regions in cortex like Broca's area, to make sure that if you're going to resect near vision related areas that you avoid causing a vision impairment in a patient or a speech impairment in Broca's area. But it was very hard to imagine how to do this kind of research in a rigorous way. Every clinician had their own way of stimulating. Some wanted longer pulses, some shorter pulses, some wanted higher amplitudes, lower amplitudes. And there was kind of an art of how clinicians do electrical stimulation. And I was trying to, these patients are rare, and I was trying to build teams that would work together. And I remember a walk with my postdocs and graduate students at the Society for Neuroscience meetings in 2013, where we were brainstorming ways of doing this. And we thought, well, the only way to really do this is to get a multi-center grant to do electrical stimulation across a number of epilepsy centers. And so the Defense Department, Darpa, announced in 2013 this call for a large team effort to try to, well, there were two calls, one for using neuromodulation to treat psychiatric disorders like depression and post-traumatic stress disorder, and the other call was for memory impairment in ideally veterans with traumatic brain injury. And that fateful day, it was Thanksgiving of 2013. The day after Thanksgiving, I buckled down and I wrote a pre-proposal. And the subsequent 5 years was the DARPA project. And I'll tell you more about that in a moment. The DARPA project for me ended about a year ago. We finished the project. We actually got a 1-year extension from DARPA. And now for me, it's a rebooting of basic science while we still continue our translational work. Well, in that little survey, I didn't show you what I was really talking about, which are human intracranial recordings. These are patients with typically epilepsy, sometimes other conditions like Parkinson's disease get implanted electrodes, but here most of our patients that we study have epilepsy. They have drug-resistant epilepsy. They've had it for a long time. And a number of clinical indicators suggest that they may be candidates for a resective surgery that would hopefully substantially alleviate their seizures. If any of you have relatives who have epilepsy, I do. It's really a terrible disease, and you would do anything you could to help these patients live a normal life. And one of the wonderful things about epilepsy neurosurgery is that many patients, the vast majority of patients who get this neurosurgery have a dramatic reduction in seizure frequency and are able to lead much more normal lives if they can map the seizures effectively in this extended period of monitoring in the hospital with implanted electrodes. And that's where we come in. So here's a picture drawn by a former student, Josh Jacobs, is now a professor, associate professor at Columbia in bioengineering. And this is his father drawing a cartoon of Josh doing an experiment with our virtual navigation games and a patient. Of course, the patient is having fun playing a game and the experimenter is having fun looking at direct brain recordings during a game. It's great. And across, well, in every patient, you get a sampling of electrodes. Now, why are they getting electrodes? Because they don't know where the seizures are coming from. I mean, the clinical team doesn't know. And so they need to put in many more electrodes and will prove to be in the epileptic focus. And across many patients, you therefore get a pretty substantial sampling of the brain. And here I show you from one study, it was a fairly large study, over 100 patients, the sampling of electrical signals. I hope that the brain is spinning for you. It's spinning for me. Hope it's not making you dizzy. The left photo shows a transparent brain with the cortical grids, and the right photo shows the depth electrodes. Now, of course, every patient's brain is different. So these reconstructions are inaccurate, not because they're inaccurate, but because we're putting all the individual brains onto a common brain. And from each of these electrode pairs, you can measure a differential voltage, and you can compute a field potential, and you can analyze the field potential while the subject plays games, does good stuff. And here's an example of the kind of games we have our subjects play. Here, the subject is navigating through a virtual town, looking for a store, and they're going to make a delivery to that store. The patient's role is that of a delivery person in this game, and the subject then hears that they delivered daffodils or whatever it was. Then they're asked to point to the next location that you tell them to go to, see if they can remember its location, and you tell them that the next package is to be delivered to the craft shop. They try to point in the correct direction, and then you give them feedback and they go find the craft shop. So there's navigation, there's virtual navigation, during which time we record brain activity. There is a memory component. We have them deliver objects in the environment, which later will ask them to remember what objects they delivered. We'll ask them to remember where they delivered the objects, or we'll ask them to remember other aspects of the experience. And we can do memory tests in a variety of ways. There's free recall, tell me everything you remember in any order you like. There's cued recall, where I cue you with a particular store and ask you to remember what you delivered to that store. So that gives you an idea of the kind of experiments that these subjects are playing. Now, the Darpa project allowed us to scale things up. Before that, we ran experiments at 2 or 3 or maybe 4 centers. We scaled up to 8 or 9 centers with the Darpa project. We were able to hire a postdoc and a research specialist at each of these centers to manage the data collection. And the University of Pennsylvania team developed the following approach to addressing the goal of restoring memory in patients with memory loss using neuromodulation. First, we would record from many electrodes while the patients perform memory games. We would then identify memory-related biomarkers, and in particular, spectral biomarkers, a spectral analysis of the field potential recordings in the neural data. We would then train a whole brain classifier, a machine learning classifier, to predict memory. And a little secret of this is it's very important that you try to predict memory not in a holdout trial or a holdout block of an experiment, but you're trying to predict memory in a future session. So a future day when the patient will come in and we'll do an experiment. Otherwise, the models don't really, I mean, they seem to be great, but they're not. And then we would search the stimulation parameter space to identify the best stimulation targets based on the decoders. In other words, can we use decoders to map out which stimulation parameters are most likely to work? And finally, deliver stimulation in real time when memory is predicted to fail, with the goal of hopefully improving memory, or at least not impairing it. During the 4-year study, we collected data from 422 neurosurgical patients, about 10 or 15 at each of our centers per year, 2,400 sessions, over 1,000 brain stimulation sessions. And if somebody asked me what was the great achievement of this project, I would say we learned how to work as a team and share everything. We shared all of our code across the 10 centers. We shared all of the data, and we publicly released everything with every publication, publicly released the data, the code, and it really developed a way of working as a team so that it didn't become siloed, every graduate student with their own experiment, every center with their own papers. What were some of the major findings? We could reliably decode intertrial variability in memory function. I'll tell you about that again in a moment. These decoders recovered brain-wide biomarkers of successful memory. We discovered that low amplitude, short duration electrical stimulation was extremely safe. It almost never elicited a seizure. In fact, the seizure frequency with stimulation was no higher, maybe slightly lower, but no higher than the seizure frequency without stimulation. And it helped us figure out when, where, and how to stimulate to try to improve cognition. Actually, it helped us mainly figure out how not to impair cognition, and occasionally how to improve cognition. We found that medial temporal lobe stimulation generally impairs memory, that lateral temporal cortical stimulation on the left side often, but not always, improves verbal memory, and that it was very important to apply stimulation only when biomarkers signaled failure of the memory system. And then finally, I'll comment at the end maybe about some of the challenges of doing biomarker-guided stimulation. We need better technology. Now, nearly all translational human research targets biomarkers of disease memory, but a common observation in our daily lives is that within an individual, memory is highly variable. All kinds of cognitive functions are variable. And even patients with severe memory impairments, such as an Alzheimer's disease or traumatic brain injury, have good days and they have bad days. And performance on their best days is sometimes not so bad. So I call that the variability puzzle. And what we're trying to identify in the brain are the neural signals that underlie intra-individual variability. Why is it that my memory works well at sometimes and less well at others? And we did one really big study at Penn in the lab, not with the patients in the hospital, where we had individuals come into the lab for many, many sessions, 36 hours of testing over 24 sessions. And in each of those sessions, they would do multiple experiments where they studied a list of words during the encoding phase. They studied words like tape, mall, dad, flood, beam, ring, camel, map, girl, 24 items per list. Then they did a period of mathematics problems as a distractor. And then they tried to recall those studied words in any order they liked. And what was unique about this experiment is that we just kept doing it over and over and over and over again. The same people got lots and lots of practice doing this experiment. I always debated when I was starting in my career whether I should go into psychophysics or into cognition. And I think I always missed psychophysics a little bit. So I tried to design psychophysics like memory experiments. And this was one of them. We were able to get so much data from each subject that we could build extremely reliable and robust predictive models of performance using all standard known variables for each individual. And we could then ask about the residuals of those models, like what is the leftover variability in memory from moment to moment? And you might think there's not that much, but actually there's an incredible amount of variability. And I show that estimated variability for each of our patients ranked from the poorest remembering to the highest remembering patients, and worst memory to best memory. I'm showing my intra-individual variability in this presentation. And it turned out that this enormous variability was best explained by performance on the prior trial, suggesting, and this is written up in this 2019 publication, suggesting that there exists in the brain some endogenous factor that underlies variability in cognition. And the best way to predict it is just look at the neighboring list, the neighboring trials. Okay, so back to the brain. What are the neural signatures of this variability? And I already showed you this, but now I'm just showing you that we can decompose that local field potential recording into frequency bands. So the aforementioned theta oscillations would be the 4 to 8 hertz oscillation shown here, if you see my mouse. And high frequency signals are sometimes called gamma or high frequency activity. And as I mentioned earlier, there are many biomarkers of good memory, and I'll just mention a couple of them here. At individual electrodes, you will sometimes see prominent theta effects where theta increases during good memory encoding and during good memory retrieval. We actually just published a paper in PNAS about this, but that is a minority report, meaning if you aggregate all the data, you tend to not see much of a theta effect. You do see a small theta effect, but what you see instead is a tilt in the power spectrum with increased high frequency activity signaling good memory and diminished low frequency activity centered around the high theta alpha band. And in this little video, I'm showing you a map, a time evolving map of the differential between good and bad memory in the brain as a subject studies a word. I'm not going to try and comment about all these regions. It's just the kind of thing you can do with these recordings with high frequency activity, which has high temporal resolution. There are also connectivity biomarkers of encoding and retrieval. There's increased high frequency activity during good memory. And you might think that that goes along with synchronous high frequency or gamma activity, but that's not the case. You do not see synchronous high frequency activity. You see, in fact, the opposite, desynchronization of high frequencies. And when you see good memory, what you see is rather than seeing increases in theta in the aggregate signal, what you see is synchronous theta. I think there was a typo on this slide. No, the point of this slide is to say that the increases in theta are not the same as the synchronous theta, and the increases in HFA are not seen in synchronous HFA, and I'll tell you more about that later. In general, the finding which we reported in several papers is that increased HFA and decreased low frequency activity go along with the reverse for synchrony, decreased synchronization at the high frequencies and increased synchronization at the lower frequencies. And here's a map of hubs in the brain from Ethan Solomon's Nature Communications paper showing some of the most prominent theta hubs that show increased connectivity predicting good memory. These same sites show decreased theta activity in the aggregate predicting good memory. As I said, the increased theta effect is a minority report seen in individual electrodes. Okay, moving to multivariate decoding. In aggregate, we might wonder, can we use the spectral biomarkers that I've described to predict future memory states of encoding or retrieval? And the answer is yes, we can. I'll show you in a moment. We can use machine learning to decode using the features of the intracranial EEG signal. And we will use that in our brain stimulation studies to try and boost memory. Now, why would you want to use these machine learning signals? Because they're potentially useful in controlling brain stimulation, because you know, for example, when memory is predicted to fail. And if you want to modulate a multi-stable system, a system that could be in multiple configurations, you want to do that when you think you can make the system better, which is typically when it's not working well. And finally, I just want to mention that these decoders often can decode different tasks. So a decoder trained on one task, you can examine its ability to model performance in other tasks. And there's going to be a common set of biomarkers across tasks and biomarkers that are unique to specific functions. And that's a whole other lecture that we won't have time to get into. Okay, here I promised you I would show you some actual data on decoding. And here are data from multivariate classification of brain activity to estimate whether or not a studied item in a word list experiment would be remembered. And so what's shown on this slide is a time course of the first 8 trials in 2 subjects. I just picked the first 8 trials, literally the first 8. And the first 8 items within those 8 trials, just to shorten the time series to show. And what you see is the classifier output probability in a holdout session, plotted from 0 to 1. 0 means you're not going to remember the item. 1 means you will remember the item. And this is the classic contrast of trying to use encoding periods to predict subsequent memory. That's the classic Anthony Wagner 1998 Science paper. And so here we're using that with intracranial recordings, applying that with multivariate methods and showing that we can reliably predict which items will and will not be remembered. The red dots indicate that the subject remembered those words, blue dots that they forgot the words. The blue dots tend to be at the bottom of the graph and the red dots at the top. And the ROC curves, which plot hit rate versus false alarm rate, sensitivity, specificity analysis, show you that in these 2 subjects, you could reliably classify memory quite well. And in 100 subjects, you can reliably classify memory in almost all of the subjects shown here in this ESIOT study. The figure on the right, I won't get into, but I'll just say that it shows the degree to which biomarkers during encoding cross-classify retrieval and biomarkers at retrieval cross-classify encoding, which is relevant to a therapy because a therapy won't know whether somebody is encoding or whether they are retrieving. Okay, the Darpa project sought to develop a therapy with brain stimulation for people who have memory loss. So the scientific question is, can we use direct brain stimulation to modulate physiology and memory? The background is that clinical brain stimulation mapping generally, but not always, impairs function. So we asked several questions. First, does timing matter with respect to variability in brain state? I have to acknowledge my wife, who's an economist, for suggesting this analysis. I thought we needed a real-time system. She said you could simulate it in a first study with a retrospective analysis of your data. And we found, in fact, in that first study that timing did matter, that when stimulation arrived during states predicted to be poor, you tended to boost memory. When it arrived at states predicted to be good, you tended to impair memory. And of course, you have to consider regression to the mean as a control for that possibility. And of course, the real test is a closed loop system that prospectively stimulates when memory is predicted to fail, and we did that as well. We asked whether the region stimulated matters, and we found that stimulation of MTL impairs, and generally stimulation of left lateral temporal cortex improves memory. And another paper that we just published with Josh Jacobs and colleagues is on which parameters are optimal for stimulation, and the aim, of course, is to develop a therapy. One little advertisement for Ethan Solomon's 2018 paper, and I won't get into the details here because of time, is that you can also use functional network connectivity to predict the physiological effects of stimulation. So I mentioned before that there's a very strong theta connectivity effect. So two parts of the brain exhibit theta synchrony and theta coherence during good memory as compared with poor memory states, both at encoding and at retrieval. And so Ethan simply asked whether knowing the connectivity between the stimulated site and the rest of the brain could predict the effects of stimulation elsewhere. He found that it did. He found that the effect was theta specific, but he also intriguingly found that it was strongly modulated by proximity to white matter tracts, which I don't think will surprise any of the neuroscientists at the Institute. Well, okay, so here was the showstopper, the study that we were working on for 3 years to set up. We created a system together with Medtronic that could record 256 channels, immediately analyze the data into the spectral domain, compute those classifiers, and stimulate up to 64 channels, up to 64 channels in response to the recorded signals. We created a closed loop system. We applied the closed loop system in a cohort of, I believe it was 18 patients, and we found a modest but highly reliable boost in memory, 18%. This study was replicated in 2 subsequent experiments. We thought we could get the effect to be larger. We had a cohort of TBI subjects. We were able to get the effect from 18% to 19.5%. Still a modest but significant effect in this TBI cohort of 8 subjects. I want to point out that there's a lot of variability across people in the beneficial effects or not beneficial effects of stimulation. So here you see the 8 subjects. The average effect is just shy of 20%. And here I show where we stimulated and the increase or decrease. And what you see, of course, in the data is that there are patients who did not benefit, maybe even slightly harm, but not significantly, and patients who benefited a lot. And each of these upper 3 patients would be statistically reliable for a within subject comparison of their data to the baseline. We did another replication, which we're writing up now for publication, with a much larger cohort using slightly refined algorithms. And we got, again, about a 20% boost in memory. So we're now seeing a consistent but modest benefit. And the hope is that we can come up with better technology to do this. Our technology is highly limited. Okay, I want to end with the vision. What would you do if you wanted to build a therapy? And here's a vision. And one of my close collaborators who helped to run the RAM project with me at Penn, Dan Rizzuto, spun out a startup out of the University of Pennsylvania to develop this technology. It uses an earpiece. Much of it has already been prototyped. And the hope is that someday soon we could implant patients for whom a boost of memory, having a 33% chance that their memory would get boosted by 40% would be worth being in a trial of such a device. So that's sort of the vision, and I'm happy to discuss that in the Q&A. So to wrap up the summary, univariate and multivariate biomarkers of variable memory function. I described those in terms of theta and high frequency activity. I spoke a bit about connectivity biomarkers and how you could build multivariate decoders, how those decoders could identify common networks for memory as well as unique components of memory that are specific to different functions. Finally, electrical stimulation can impair or improve memory to the extent that it modulates these biomarkers. And this is just a couple more points. We learned not to stimulate MTL. We learned not to stimulate at too high an amplitude or too low an amplitude. And we learned not to stimulate when memory was predicted to fail. What was the most important thing we learned? We learned that this is a really hard problem, that it's really hard to boost memory reliably with brain stimulation. And maybe that's for the same reason that it's hard for Fed policy to boost the economy, because the brain has a mind of its own and it wants to do its own thing. And unless you whack it hard enough to break it, your ability to kind of nudge it where you want is somewhat limited. So I still think I'm hopeful that biomarker-guided stimulation will offer therapy for the future. We need better technology and we need smarter algorithms. And I want to thank my team that did much of the work that I described to you, and I welcome your questions.
Thank you very much, Mike, for that presentation, for describing this very broad and interesting open project. We have a question really about the biomarkers themselves, and let me help with that framing a little bit. I wonder, we don't have a lot of time, but I wonder if you might answer this. The different subjects, different participants had their electrodes at different places. And I'm wondering whether there was much specificity in talking about biomarkers. You didn't talk a lot about the position of the electrodes in individual patients. And I wonder if you might say just a little bit about what the biomarkers were and to what extent they were place dependent.
Yes, right. Well, I mean, I think that, of course, everybody has strong priors on where you will find biomarkers of certain things. So yes, you find place cells in the hippocampus, in the human hippocampus. You find grid cells tend to be in the human entorhinal cortex in the single unit biomarkers, consistent with the animal studies. And with the field potential recordings, you find particularly strong effects for memory in bilateral hippocampus, for verbal memory in left lateral cortex, left prefrontal cortex, and parietal areas. And that's the standard network that has also been defined with fMRI. If part of your question was, does it depend on where a particular patient had their electrodes implanted by the neurosurgeon? All I would say about that is that this is an incredibly heterogeneous population. There's a classic study by Ogeman of Broca's area. We all know where Broca's area is, but if you look at these patients and you literally identify Broca's area with disrupting speech by stimulating the brain, it's really all over the brain. And it tends to cluster where you expect, but it's really all over. And so there's huge variability. So I guess I would just summarize: a lot more variability than the literature suggests, number one, and a lot more distribution than the literature suggests, number two.
Okay, well, thank you. You know, there is a question just asking you for a general thought. I guess in the age of Neuralink, we're all wondering about what is the best resolution and position and nature of the signaling. So if you could just take one more swing at that, though time is quite short. But if you could take one,
yeah,
that was the question.
Absolutely. So Brian, yes, it's a great question. And so we actually did a study. We wanted to know how many electrodes would we need and how widespread would they need to be to decode reliably. Turns out that to decode reliably, it's much more important that you have electrodes in widely distributed regions, and you really need to have 128, at least 64, ideally 128 electrodes. We did it in powers of 2 distributed. And I think the size of the electrodes is, you probably don't want to go too small, because I mean, you could record single units and it's an exquisite signal. It doesn't stay for very long. It doesn't last that long in human neurosurgical patients, because you can't keep the neurons for that long. But you get a myopic view. And what you really want is a less myopic view of brain function. And so you really want to survey the brain. And so I guess I would just say you want to survey the brain at a lot of regions, at a broad set of timescales, and you want to look at the connections between those regions and the activity within them.
Well, thank you for that. Although there are more questions, I'm sorry that we have to bring this to an end, but I'm looking forward to bringing you out here and having a much more extensive conversation.
Thank you very much.
Thank you, Mike. That was great. I've been asked to mention to those of you who are addicted to Twitter, as I find myself sometimes, to follow this event on at Stanford Brain, and that's, I guess, for mostly younger people. I don't know. And then I also would say we're going to take a break now for 20 minutes, and we'll start promptly at 11, and look forward to seeing you all back then. And thank you very much for your participation.